My first question is, how did you get into it?
Oh, Anna. Oh, well, um, I was a medical student at UC San Francisco, and then an intern. And when I was a medical student finishing clinical work, I realized that maybe I would like to give research a try. I had done a little research earlier in medical school, and so I signed up for after my medical internship to go to the NIH. And and this was part of the U.S. Public Health Service. It was during the war. Um, this group of NIH researchers were sometimes referred to as the Yellow Berets. There was one uniform that we shared between us. In any case, so I was signed up for that to begin in 1975. And during my medical internship at San Francisco, I was scared. I was really scared because I was going to arrive at the NIH and I didn't really know any neuroscience. And I signed up for a place in Ed Everett's lab in the Laboratory of Neurophysiology, actually because that lab description sounded like they were interested in the whole brain activity, as opposed to a synaptic protein and so forth. So during my internship year, being so scared in anticipation that I would arrive there and embarrass myself, my family, my school, I purchased, I don't know whether some people will remember this, there was a 3-volume set called The Neurosciences. Later there was a 4th volume. And this was put together by a group called the Neuroscience Research Program, MIT Press, a number of luminaries who later, I think, really were instrumental in founding the Society for Neuroscience. So there was no Society for Neuroscience. There were neuroanatomists and neurophysiologists and neuropharmacologists, so forth, but there was no Neuroscience Society. And I bought these 3 volumes and read all of them in the evening during my internship, so I was pretty tired. In one of the volumes was a series of papers edited by Pittendrigg, Colin Pittendrigg. There were papers by, I can't remember them all now, but Truman, Strimwasser, Pittendrigg himself, maybe Jacklett, on the neurobiological basis of rhythms. And one of those chapters was by Bob Moore. And there he described experiments in which implicated this nucleus above the chiasm, as the it was known as a suprachiasmatic nucleus. And I remember there was a big picture in this book, a Nissl stain, that showed the location, and you could see these beautiful nuclei there. And then I went off to the NIH, but I remembered it. I had a bit of a photographic memory that time. And so I set off the NIH, and actually what I was supposed to be doing was recording from the substantia nigra in monkeys. Ed Everett's was a neurophysiologist, like Vernon Mountcastle and others, and that was my assignment. But I also met there a guy named Frank Sharp, who was doing physiological studies using a new method that hadn't been published yet, that Lou Sokoloff had invented and developed, called the deoxyglucose method, which is now famous, as a method for measuring energy metabolism, that is fuel consumption, in the brain using autoradiographs. So it was a demonstration of regional fuel consumption, beautiful autoradiographs, and of course the modern day equivalent of this are PET scans with fluorodeoxyglucose. And so this looks interesting because I have kind of an anatomical bent, and they were pretty brain sections. But I noticed on them, when they cut through the whole brain, that through the hypothalamus there was a huge crack. So it went from maybe the anterior commissure all the way down, and it was cracked and deformed, or the hypothalamus was blown out. And I said, that doesn't look so good to me. I mean artistically. So I went down to Sokoloff's lab and spoke with the the technician there, the very able technician, who had worked for Sokolov for many years and had been cutting the brain sections. And it turned out that they were, because he was a neurochemist, used to measuring intermediate metabolites that were short-lived. The brain would be taken out and maybe even in the skull would be frozen in liquid nitrogen. It was very cold, and things would crack. And the technician said, no, that's the way it looks. That's the way we get it. But I think striving for some artistic perfection. Frank and I tried to figure out other ways to freeze the brain so it would look a little nicer. And we came upon a method that was gentler. It was cooling the brain in isopentane, cooled to about -20 or -30 degrees, which I guess is used a lot now in all sorts of in situ and so forth. And the brain sections came out much better. So I felt happier. Then I was cutting through the brain and was much happier with the atlases of the brain. Now, since I was no longer an intern, I also had some free time to do various things. I didn't have much of a musical education, so I thought I would learn something about music. And I would go to the Library of Congress, where they would have quartets in residence. Very pleasant. Forget, maybe once a week, something like that. And it was at one of these, with the Budapest String Quartet, that I met Hal Gaynor, who, of course, many will know, as in that time in the 70s was really a pioneer in neuropeptides and distribution and processing, synthesis, and so forth of neuropeptides. And in the intermission we would talk. It was an agendaless talk. And I told him what I was doing, and he mentioned, and he said that, he had a good experiment, actually, which was related to supraoptic nucleus and pituitary, and looking at secretion and energetics of secretion. I said, gee, I could really make good sections through the hypothalamus now. And so we agreed to do it, and I did a pilot experiment, and for the first time to do this, I cut serial sections through the hypothalamus. The animals were in a light-dark regime. I sacrificed them in the middle of the day, and I looked at the sections, and in a rare moment, there were two dark dots above the chiasm, meaning two areas with increased glucose utilization. And I remembered the Moore paper, and I said, those are suprachiasmatic nuclei, and gee, after all, those are supposed to be maybe the locus of an internal oscillator or pacemaker. And I think it must have taken 20 or 30 minutes of a discussion with Hal, which was always fun, to consider what the experiments might be that we can all think of now. And it took me 2 weeks, I think, total, to do the experiments. Then we wrote it up. I hadn't been at the NIH very long. Sent it to Science. And the review came back suggesting that we add a scale bar to the picture. And I said, this is wonderful, this rhythms work. Later, actually a couple of years later, we actually were able to do Hal's experiment, which also turned out to be important for deoxyglucose. And the part of that, along with others in Hal's game, Marina Mata, Dave Fink, Carolyn Smith, and Sokolov Lab, really gave insight into what the 2DG was measuring, which is still true for PET today. And that was another really nice paper. So, I finished my time at the NIH, and I went back and did a neurology residency, but I was hooked. And, I was a resident from 1978 to 1981, got my first job, and I was able to continue doing the experiments I had thought of. So that's how I got hooked. The lessons I learned were actually useful to strive for perfection, because in this case it was the difference. just to make it right, make sure it's right. You know, look at the original data, looking at my own autoradiographs, making sure you associate with smart people, certainly smarter than yourself, and taking some nights off for some music, I think. So that's what I learned, which I've tried to remember since then. So after that, I was hooked.
Moments that were important, where you did things and the field changed. We have to think in terms of the context of the times, you know, in the late 70s, 80s, how we were thinking of the circadian system, input, black box, output, formal properties. And when CFOS stimulation of light came along, there was gene expression.
Yeah, I mean, there had been papers, you know, again, well, with the, you know, again, you see, with the deoxyglucose as a measure of functional marker, there was a wide interest in this as a general functional marker, and that it would just mark active cells. And of course, once people realized that was really a gross oversimplification, because really what it was measuring was energy demands, and so those depended on surface to volume ratio. It depends on whether the cell is being stimulated, inhibited. It's a different kind of measure. And then sort of it cooled off. The same with CFOS. CFOS started out, people had written, this is a measure of neuronal activity at a cellular level. And there was great excitement on this. And of course we had read those things. I had a buddy, by now I was at UMass. I had a buddy, Neil Aronin, and the two of us thought this would be an interesting thing to try, whether in fact we might find photoinducible gene expression. And I guess it's fair to say that CFOS was the first photoinducible gene. It really was in the vanguard of where we are now. And as we were doing the studies, we were very excited about it. And I can't remember, somebody had told me that Ben Rusak was also working on this. I forget how I came to know that. So I called him. And I said, is it true that you're working on CFOS gene expression as a marker in the SCN? Yes. So we talked about that, sort of initially. But then, after all, it was great, and we had decided there was a Gordon conference coming up, and we decided, why don't we sort of petition the two of us to give a little thing in a special. I forget who the chair of the Gordon conference was at that time. The conference would have been 1989. We could look it up. And anyway, we were permitted to do it, and we, you know, I mean, it's hard to imagine what that was sort of like then. I mean, now we see these, well, we see these things all the time, but there was a photoinducible gene in the SCN, in the ventrolateral part, that was gated. There was the photoinducibility was phase dependent. It was on an input pathway. It looked like we were looking at an input pathway, and it also gave us, of course, a marker for stimulating the SCN. And it was well received. It actually was well received, and I think that year I was elected to be the vice chair of the next Gordon. So it was well received. But again, I was I was working on this SCN level of things. We were really working on this tissue level sort of thing, and of course it was years from the beginning before Martin Ralph and Menaker had done the transplant experiments, that I think everyone was really quite satisfied that this was a oscillator, that was a pacemaker that was both necessary and sufficient. Now I should mention that those, because it's another thing I'm proud of, is that those transplant experiments obviously used fetal tissue, and the basis for the fetal tissue having a functional clock was, I think, supplied by Steve Reppert and me some years before, where we'd used the 2DG method in a pretty neat experiment, I think, where we showed that the oscillator in the SCN was really oscillating in utero before and synchronized to the light-dark schedule before the animal was born, before the animal really had any knowledge of light and dark other than through maternal signaling. And of course it's interesting in retrospect because we knew, this was Bob Moore's work, that the synapses really hadn't developed at that point. It was really a syncytium of cells, and one could have guessed then that perhaps the oscillator machinery was really intracellular rather than depending on synaptic connections. So already there was I remember thinking, gee, the oscillatory machinery is much simpler than this complex nucleus, which only develops later, maybe for entrainment and output and making it a clock that was useful. But the basic nibble of oscillatory action was was from a much simpler homogeneous substrate.
Ticking was already there.
The ticking was already there. The things that have really been enjoyable about this field and this area, I would say, which I tell people, is personally, I've been able to continue working on, a millimeter, it's a millimeter long or less, and this same problem, this same tissue, and yet throughout this time learn all these different approaches, techniques, methods. So I can do all this learning in neuroscience and still be working on my same place. So here I learned about energy metabolism, cerebral blood flow, I learned about development, I learned about gene expression. Since then we've done some stuff with electrophysiology, and so I could always be learning and applying new tools and still be working on the same problem and asking more questions. We went out to dinner, my wife and I went out to dinner once with a wealthy friend of ours who's made a lot of money and retired early, very successful, and he asked, what I was doing and so forth, and I described it. He said, well, if you're working late at night, do you pay yourself time and a half? And I said, well, no, I said, it's more like being an artist. And he understood that better. And then he said, you're really lucky. I said, I'm lucky. I thought you were lucky. No, you're really lucky because you've been able to sort of continue, be passionate working on this kind of area and learning all the time. And that was true. So that was the one thing that really has been fabulous. And the second thing that's been fabulous about this field at these meetings is the diversity of people and personalities and organisms, all gathered together, sort of focused on the same problem, using the same kind of logical reasoning, using the same kind of base, but then synthesizing and synergizing different organisms. I mean, right from cyanobacteria to people, and different disciplines, microbiology, molecular biology, clinical psychology, neurophysiology, so forth and so on. And yet we're all gathered together sort of focused on this problem. I'm a neurologist, and the epilepsy meetings and so forth are not like that. And the range of personalities, I mean, a meeting with a microbiologist or a molecular biologist or fungal geneticist and a neurobiologist and a clinical psychiatrist is very interesting.
And they have the same formal properties.
And they have it's the same formal properties. And so it's really given me an opportunity to meet people that I just wouldn't have met before, and it's been spectacular. It's really spectacular. We've had a good time. And we still are, Anna. Yeah. And for, it's worth saying, probably others have said this, for those of us who went into neuroscience or neurobiology, where our question was, we'd like to account for behavior, the substrates of behavior. And really what we imagined was at multiple levels of organization, at the molecular and the cellular and then the tissue and then the whole organism, one would add pieces to that, and so various parts of the system and the function of the system would emerge at each of those levels. And I don't think there's a better system than the circadian system in neuroscience that has been getting at that. And it's just been fun for me to work at different levels as well. So I consider myself, for the record, this is the record, I consider myself extremely fortunate.
Can we go back to after your deoxyglucose and CFOS, then came your dissecting the SCN into functional bits that surprised people just because you cut it in the wrong direction by mistake on the Tuesday. You found out something about?
Well, yeah. Well, the interesting thing about those kinds of experiments were also that once David Welsh and others that followed had shown that the basic oscillatory unit was cellular, and people were chasing the molecular genetic basis for this. As a neurologist, I know that once you have up to, well, no greater than 20,000 neurons gathered together, presumably you can do much more with those. And wouldn't it be interesting if there was some plasticity or flexibility related to that? And the original experiment we had wanted to do was we were doing some experiments actually with FOS, but they depended on having a slice, and they depended on stimulating the optic nerves. So we started practicing making horizontal slices with the nerves attached. They were really quite nice. They were hard to look at initially because we didn't really, we had been looking at coronal sections so it was kind of hard to figure out where you were, but after a while, if you stuck with it, we could tell, and you could see them just as well in the horizontal slice as we were used to in the coronal slice. And a postdoc, Anita Jacoda, who was a very dedicated person and quite persistent, had been working on recording multiple unit activity. She hadn't had, at that point, as much background in circadian biology, and we had successfully recorded from coronal slices beautiful multi-unit rhythms of, looked like Gillette and those kinds of things. So we went to the horizontal sections. She tried, and she said, I asked her how it was going. She said, well, it wasn't working very well. And I remembered my lesson that I mentioned, look at the raw data. I said, well, let's look at them. Maybe we'll get some clues. And what I saw was that they were not so good because they had 2 peaks instead of 1. So actually I didn't say anything to her. I said, well, why don't we do some more of these? And so she did some more of them, and then we put them all together, and there were 2 peaks instead of 1. And I said, while we're at this, why don't we entrain the animals to a different photo period and then record again? And again, we saw that one of the peaks changed and the other didn't. And I said, while you're at it, why don't we give glutamate, in the evening and in the morning, and then we can see we can move these peaks separately. And then I said, I wonder whether these are really the representation of the phantom, mysterious dual oscillators, morning and evening of Pittendrigg and Don. And so that was really very exciting. We sent it to Nature. I thought it was a pretty good paper. It had 3 reviews. 2 of the reviewers liked it with not much change. The other reviewer said that you haven't really shown that these are oscillators. They're really oscillations. I mean, you've moved them independently and so forth, but you haven't shown that they free run independently. So they're oscillations. And with that, the magazine declined. But we ultimately sent it to Nature Neuroscience with the reviews, and they said, well, maybe you could just answer these reviews or change them. And so we sent it to them, and then within a very short time, like I remember from decades before, we were able to get it published, and I think it kind of brought them back. It brought morning and evening back a bit. They had been around, obviously. Helena Ilnarova, others had sort of been working with this idea, but I think it was the first substrate that fit the description, the phenotype for the morning and evening within the SCN. And of course the problem with it was that we had no cellular resolution with it. But at least it opened the door, and now there's great stuff being done on this topic.
Because that was more in the pineal with Helena's work.
Right.
That people accepted, okay, it's the signal for seasonality and so on, but in the SCN, that was really
Well, and
important. Coding.
Right, and we knew we had worked with Helen also on using CFOS in sort of another nice application to show that actually the SCN was keeping time. The photo-inducible subjective night, as measured by CFOS, was decompressing and compressing. So we knew the SCN was doing this, but there was no way to pull it apart. And of course, we still don't know. I still don't know, to tell you the truth, why cutting it horizontally has exposed those or revealed those. So there's a lot we don't know about what we measured there. But the field has taken it up and gone great guns with it as well.
As the new techniques come along, then you can see these pictures of oscillating glows of different parts of the SCN, and you couldn't do that at that time.
Well, one of the things that's kind of sweet is, for us oldsters, not counting you, but for me also, is pulling out some of this old literature that perhaps that may not be in PubMed or so forth, and then reopening it. And of course now that we have these tools that you've just described, the formal stuff becomes even more useful, not yet less useful. So that's what I've been saying. People have said, well, now that we have all the molecules and the things, we don't need that. No, actually, that now becomes really helpful. And this splitting business was sort of an example of that, because we had the tools. Horacio de la Iglesia, who was I should mention, on the Two Peaks, Anita Jacoda, who's now assistant professor in Hyderabad, did that work, and I still am in communication with her. Horacio de la Iglesia was in the lab, and somehow we agreed we ought to take a look at split animals, and I remember I had made a prediction that they were the two oscillators of the split condition of Pittendrigg and Don were going to be the left and right SCN. And I said that because soon after the 2DG, returning all the way back to 1977, I met Pittendrigg. I went out to Hopkins Marine Station. I remember showing him, it was not published yet, the 2DG work. Gene Block was there, and Gene Block and Terry Page were both there as postdocs. And so we were poring over this. And then afterwards, Pittendrigg wrote me a little note, which I saved, which said, very interesting, nice to see you, and so forth. And I have a good experiment for you. And the experiment was the splitting, because he had already was wondering about this. And of course, in the mollusks, there was already an example of how the left and right eyes, were two oscillators. And I tried that experiment actually later with Nicholas Mrzowski with 2DG, and there was intermediate glucose utilization, probably because there's excitatory activity on one side and inhibitory on the other, and it would just be equally metabolically expensive, perhaps. But it didn't show anything. But Horacio did these experiments, and then he, in a nice circle of life, he told me the autoradiographs were coming out, and I said, well, you should look at them first. And I think he had must have had the same emotional impact as when I first saw those 2DG ones, because you could see right away that there was only one SCN on. And then we did more experiments with BMAL and so forth. But it was very satisfying, because we used some data that had been 30 years old, really, and kind of brought it back to life. So I've been fortunate.
One thing to do with the story of the interviews is to go back to the papers and say, this was there, you just had to reinterpret it with new methods, or you had to do new experiments, but the structures were there. I mean, that's why putting and down papers was so important, even though the geneticists thought.
Well, and it's why it's so important to actually read the papers.
And now, especially
Now, because
they don't know their rules. Most of them.
well, and there are a lot of papers, and if people are too busy and so forth. I mean, there's proliferation of stuff. But you actually have to read them and think about them and remember, and then call them back. I have to say that I still read current contents. The book version. I shouldn't really be admitting this now, but I still read them. First of all, they're very portable, so when we go out shopping or my wife goes out shopping, I can stay with her and comment on the clothes and also have my current contents. And I look through this current contents, and it's like reading a telephone book, as you know. But the beauty of it is, as opposed to a keyword search of some kind, is I sometimes find things that I didn't know I was looking for. Or sometimes on a daydream, a title or two, as I look through, somehow calls forth something else. The yield is extremely small. I agree.
But it's the
but it's there, and it's the stuff I didn't know that maybe I would need to know now, or something I hadn't thought of, or a clang association of some kind. It's just not the same. It's not very efficient, and the keywords are the efficient way to do it, but maybe it helps on having these kind of moments of ideas, these ideas, which for me is the most fun of this. I mean, what is still great about this field is this liberal education idea. It's really the tradition that, and it comes from the Gordon Conference in the early days of the SRBR, that all of us would be hearing about the rest of our work. That I had, it was appropriate for me to learn about Arabidopsis. It was appropriate that I should be learning about Neurospora and the conceptual thing. And I hope as we've grown and with knowledge, it doesn't start to fracture things. That's the big worry, because that was really a special feature. So I hope it continues.
It still continues in the Gordon because there's only one session.
And that's the beauty of that. And
a neurologist who knows about Arabidopsis.
Crazy. That's
Nice
the liberal.
geneticist.
That's a liberal education.